Our first presenter is Dr. Gowthami Arepally. Gal is professor of hematology at Duke University and runs our platelet research lab. She got into complement and its role in blood through her work in heparin-induced thrombocytopenia, and we asked her to speculate about the role of complement in uterine hemorrhage. Thank you. Thank you, Gal.
Welcome, everybody. It's good to be here. Thank you, Andy and Ken, for inviting me to speak. I think the operative word for my talk is speculate. So I will be talking about the role of complement in uterine hemorrhage. So Andy came up to me a while back after one of our hematology conferences that she attends, and she says, Gal, we're putting together this fantastic program on uterine hemostasis in the spring. So, do you know anything about complement's role in uterine hemostasis? And I'm like, no, not really. She goes, well, that's wonderful. Why don't you talk about it? And like the good sucker that I am, I said, sure. And after hours spent looking into it, this is what I have, which is not very much, but I do think there is, I will show you some of the information relating to complement's role in uterine physiology, and we certainly know that coagulation plays a prominent role in menstrual bleeding, and so it is really not that much of a stretch to connect the two dots of both complement and coagulation, the role of these two systems in uterine hemostasis. These are my disclosures. And my work with these companies has no bearing on today's talk. And this is the outline of my talk. So I will be giving a brief overview of complement. Do you know where the pointer is? Is this? What? The pointer on this thing?
This one is supposed to be the pointer.
Oh, okay. So, it's very light, that's why I couldn't see it. So I'll be reviewing about the complement system. I will talk about complement and coagulation, how they're both partners in host defense, and the numerous interactions that complement coagulation have. And I think this is a hot area in research, and it's only going to be much more expansive in the years to come. And how they have they are individual functions, but really have a lot of overlapping and reinforcing effects in both health and disease. And then I will, the second half of my talk will be about complement in uterus and complement's role in host defense, role in reproduction very briefly, and talk about what minimal data there is on complement and uterine hemostasis. So I think all of you know in the audience that complement is a very vital part of the innate immune system. And in its role, it interacts with virtually all the cells that are involved in immunity. And this includes its role in opsonization and tagging bacteria and pathogens for clearance, as well as lysis. Its very important role in alerting neutrophils and other cells to pathogens. It clearly has a role in inflammation, as we know in a number of diseases associated either autoimmune or infectious disease, where complement activates the inflammatory pathways and individual cells through cellular receptors. It also is very vital as a disposal system in physiology and homeostasis. So it clears and solubilizes immune complexes and gets them cleared from circulation. Is also very important for clearance of apoptotic cells. And how we got into complement was our work in understanding the immune response in heparin-induced thrombocytopenia. So it has a very vital role in adaptive immunity. So an impaired complement system affects the adaptive immune response and its ability to recognize pathogens that it needs to make antibodies to. So it promotes humoral responses as well as T cell responses. It's critical for self-reactivity in eliminating B cells with self-reactivity, and vital for immunologic memory. So what are the parallels between the coagulation and complement system? And there are a number of them. And at a broad, if you take a back step, they're both highly conserved pathways that mediate host defense. And both systems have co-evolved over millennia. They're both structurally and functionally similar in terms of triggers, amplification systems, as well as inhibitory mechanisms. And I will touch on that in a minute. And they're both regulated, or they involve serine proteases that are enzymatically activated from inert precursors to active enzymes. And their regulation is their activity is tightly controlled. So the complement system, the triggers for the complement system are divided among the three pathways: the classical, lectin, and alternative pathway. The classical pathway, its main effector is a C1q complex, and it's activated by immune complexes. The lectin pathway is activated by charged surfaces on pathogens, and its critical regulator is MASP2 enzyme, and the alternative pathway is involves an amplification loop involving C3b. And the coagulation cascade, the triggers are known to many in the audience: tissue factor, which initiates the extrinsic pathway, and factor 12 and kallikrein, which are involved in the intrinsic pathway. And all of these activators converge on regulatory enzymes, that the critical one being for the complement system being C3, factor 10, and then activation of those enzymes then leads to activation of effector enzymes C5 in the complement system, and factor 2A or thrombin in the coagulation system. And then these are the mechanisms by which the inflammatory pathways or thrombotic pathways are activated. They both have amplification systems. So the complement system has amplification where the C3 can go back and activate, and primarily it activates the alternative, which serves as the amplification system, much like the amplification system we see in the intrinsic pathway with prothrombin going back to activate some precursors. And then both systems have regulators. So factor H, factor I, and membrane-bound regulators, CD46, CD55, CD59, these are all enzymatic regulators that inactivate complement degradation products. And similarly, we are very familiar with the regulators of coagulation, primarily being antithrombin, tissue factor pathway, and protein C, protein S systems. But there are also critical differences. Unlike a coagulation proteases, factor 10 and factor 2, the central enzymes, C3 and C5 convertases, are very inefficient enzymes. And then you also have complement split products, C3A, C5A, that activate receptors and mediate inflammation without having significant enzymatic activity, without any activity. And then you have a number of interactions between the two systems where you have mediators on the coagulation side that can activate the complement system, and then you have complement enzymes or effectors that activate the coagulation system. And I'm not going to talk about all of these interactions, but these are being recognized increasingly more and more as tools and techniques and interests have evolved in these two systems. So that's complement and coagulation. We also have complement and how it affects other pathways important in hemostasis. That includes platelets and fibrinolytic pathways. So on platelets, platelets can activate complement or interact with complement. So platelets through GP1B and C interacts with C3 and transports bacteria for clearance. Chondroitin sulfate released from platelets also activates complement, and then phosphorylation of C3b is known to prolong its half-life so that it becomes more active. And complement has a lot more prominent effects on platelets than platelets do on complement. So what complement does is it activates, C3a activates platelets and promotes adhesion and aggregation. C3b can bind P-selectin. Remember, this is one of the key mediators of the complement system. And then the terminal membrane attack complex consisting of C5b9 gets incorporated into platelet membranes and then induces platelet prothrombinase activity and microparticle release. And that is actually a very potent mechanism which is thought to really contribute to mechanisms of how complement promotes thrombosis in both arterial and venous thrombosis. And then there are interactions of complement and fibrinolysis, which is mediated through generation of plasmin. So when a clot gets formed and you have incorporation of plasminogen into the clot, and tissue TPA, tissue plasminogen activator released from endothelial cells converts the plasminogen to plasmin. And plasmin actually inhibits C3 convertase and C3 convertase to release C3a or C3b. But what plasmin does is it bypasses this central enzyme and directly activates C5. So plasmin can activate C5, leading to the generation of C5b9 complex. And so this is thought to be a bypass mechanism in which plasmin has direct activating effects on either cellular lysis or clearance of debris, and that is thought to be one of the mechanisms. So to summarize this part of the talk, I think I've demonstrated that there are numerous points of intersection between the coagulation the complement system and coagulation, platelets, and fibrinolysis, and that there are similarities between the coagulation and complement systems that are reinforcing each other. And then there are vital platelet-complement interactions that promote not only pathogen clearance, but both coagulation and inflammation. And then fibrinolysis can bypass earlier activation steps in the complement system and directly generate C5a, which not only attracts neutrophils, which promote degradation of cellular debris, but it also produces membrane attack complexes that can lyse cells and also promote clearing. So that was all about the background that we do know something about. Now entering into the unknown territory of complement in the uterus. So what we do know is that there is a physiologic role for complement, particularly with respect to host defense in terms of infection and inflammation. And we know that complement dysfunction is very apparent in certain disease states in pregnancy, and so we know that complement is involved in certain pathologic diseases involving the uterus. But to what extent does it promote hemorrhage is an open question at this time. So what we do know about complement in the uterus is that there are cyclical changes with complement and complement regulatory proteins. So C3 factor B is actually synthesized by glandular epithelial cells of the secretory endometrium. There is increased expression during the luteal phase. And there is also DAF and CD59 that's present in endometrial glands that's also expressed during the secretory phase. And then there are other proteins, CD46 in particular, that does not undergo cyclical changes, although CD59, CD55 do. And I did not remove the other two here. So what do we know about complement's role in host defense involving the uterus? So there have been natural experiments in which human beings have been born without certain complement factors, and particularly deficiencies in the terminal complement protein pathway beyond C5. So defects in C5 or C6 or even factor I, which is a inhibitory protein. These have been associated with recurrent meningococcal infections. And what's really interesting is that in the case reports, especially in the two that are featured here, in this one patient who had factor I deficiency, she had recurrent meningococcal infections, over 20 episodes that were all timed around the perimenstrual period. And so clearly a link between complement and menstruation in this patient, and its role in protection, because the patient was developing meningococcal infections. This was also described by Ralph Snyderman from our institute, who studied a patient who with C5 deficiency who had, once again, recurrent gonococcal infections that were also very, very closely linked to her menstrual cycles. So very striking. Yes. And this is another study in which they actually studied total hemolytic complement activity. That's a CH50. It's not showing up very well here. And that's what's shown in here in triangles. You can see during the proliferative phase of the menstrual cycle, levels are low of the CH50. And this mirrors the anti-gonococcal activity of the serum. So when they took the serum from patients at this time period of their menstrual cycle and then analyzed its effects on gonococcal clearance or lysis, it was very low. But the activity, the anti-gonococcal activity, improved mid-cycle, as did the CH50. So this is kind of an indicator that there are cyclical changes that are happening that can be actually measured through various mechanisms throughout the menstrual cycle. And then there's this one article that talked about changes in gene expression in menstruation. And so here what they did was they took endometrial samples and collected, that were collected by biopsy on days 2, 3, and 4 of menstruation. And some of these people had abnormal menstruation, heavy menstrual bleeding. And they looked at differential gene expression on days 2, 3 through 4 of menstruation. And they found that the largest number of differentially expressed genes were between days 2 and 4 of the cycle, and that they clustered around a lot of different families, primarily immune cell trafficking, inflammation, which suggests some connection to complement, cell cycle regulation, and extracellular remodeling, and complement. This is cells as opposed to humoral, and so the immune cell trafficking as well as complement and coagulation changes that were differentially expressed. And this is the top 20 genes in their in this study where they showed differences. And as you can see, not surprisingly, there was a 20-fold change in levels of fibrinogen A and B that were present between days 2 and 4. And then coming in at number 11 was complement component C4b binding protein. And so that was a 5-fold change in difference between days 2 and 4 of the menstrual cycle. So what we do know for sure is that complement has a role in thrombotic disease in certain disease states involving pregnancy. And so these are studies that have clearly shown that patients with antiphospholipid antibody syndrome, their antibodies activate complement, lead to C5a that binds to complement C5a receptors on neutrophils, which then upregulates tissue factor activity and then leads to oxidative damage that ultimately contributes to fetal death. And we know that is a truly pathologic condition. And we are increasingly seeing now reports of how HELP may be a complement-mediated disorder, and certainly atypical HUS. Both of these are triggered in pregnancy, and there's increased complement activation, and which compromises the placenta and leads to growth restriction and or demise. And the only study that I found where complement has been directly implicated in uterine hemorrhage is this study where these investigators looked at histology samples of women with postpartum hemorrhage of unknown etiology. And what they did here was they stained the myometrium here with regular H&E, and this is on the leftmost panel here is healthy pregnant myometrium. This is postpartum myometrium, and this is postpartum hemorrhage myometrium. And clearly, histologically, you can see the differences. And what they looked at was C5a receptor, and they looked at other inflammatory markers as well. And the one thing that showed prominent differences was increased neutrophil trafficking, as well as increased C5a receptor that was present in the myometrium. You can't see it here. There's no C5a receptor staining here in pregnant uterus or myometrium, and then in postpartum, but markedly increased in the postpartum hemorrhage. And so what they propose is that this could be a type of either residual amniotic fluid or fetal materials that activate complement, that lead to mast cell activation, or neutrophil trafficking through chemotaxis from complement activation, that then leads to postpartum acute myometritis and uterine atony and postpartum hemorrhage. And this is all I could find. So I think what is safe to say is that there's definitely complement in the uterus, no question about it. And its dysregulation is really problematic for host defense and likely for uterine physiology. So we know that there are cyclical changes of several complement proteins, and we know that complement is implicated in certain disease states, including HELP and atypical HUS. And we now have some preliminary data suggesting that it has a role in postpartum hemorrhage. But its role in healthy and pathologic menstruation has not been well studied, but clearly implicated. And I think there is no question that this is an area that's ripe for investigation. And if there are any young investigators looking for pilot projects, this would be one if you do have samples and or labs that are involved in complement nearby. So that's all I have to say. Thank you for your attention.
Thank you.
We're going to do questions afterwards.
Yeah,
Well, we have time for a few questions right now.
Oh, okay.
the association between the biochemistry of complement and the biochemistry of coagulation have been intriguing for a long period of time. And there's been a focus of coagulation investigators on coagulation and complement investigators on complement, but it's been very rare for a coalition of complement coagulation investigators to get together to try to see whether the hypotheses that have been generated have merit. And it strikes me that if I were starting my career again, which I don't intend to do, that it would be very reasonable to consider animal models to pursue this, because you can't wait around for a pathologic event, if you like, to study these kinds of things. Are there good murine models for complement? There's plenty for coagulation. And so that it would seem to me that with knockouts and whatever, there would be a fertile way to investigate what the likely physiology would be on one or the other process using such an approach.
Yes, and that's a very good question and a very good point. And there are plenty of murine models of complement deficiencies. And for the most part, they work okay. But human and murine complement systems are so different that it's not a given for any particular pathway that the murine system is going to model the human system. And I'll just give you an example from something we know very well. We spent almost 6 months in our lab pursuing a murine knockout of CD21 to show a CD21 is complement receptor 2, and it's uniquely expressed on B cells and follicular dendritic cells. And so we had the hypothesis that our antigen was activating complement and binding to B cell CD21. So it was simple to do that. We just went to the mouse model of CD21 and we did our studies. It did not work. You do the same thing in humans. You bind up CD21 and the antigen doesn't bind. But in mice, it does bind. So there's something with each complement protein that may or may not be modeled in mice. And so you just have to, I think it all depends on which protein, which pathway, and what you're trying to do.
How about in the converse, in adventitious, and from a scientific standpoint, human pathology with complement deficiencies, are there associated coagulation disorders that one could identify?
Yes, yes. And so one of the neatest talks that I went to at one of the complement meetings was on the role of the C5a receptor, which was one of the last slides that I showed. So the C5a receptor is turning out to be pretty interesting from the complement coagulation crosstalk. So mice that have a C5a receptor deficiency are protected from certain types of thromboses that occur in atypical HUS. So they have mouse models of atypical HUS in which factor H is dysregulated and overactive and mimics the kinds of thrombosis. So the mice have retinal thromboses, they have thromboses in the liver and the portal vein bed, and it looks in terms of similarity, it looks similar to some of the human diseases, the human thrombosis that you can see. And if you knock out C5a receptor, the thrombosis is gone. So there is some compelling evidence that, yes, C5a may be, and neutrophil C5a receptor may be one of those critical intersections between coagulation and complement. Sorry.
Hi, Julia Coleman, Denver. Thank you for a fantastic talk. I've been following a lot of Chuck Esmond's work in trauma patients on this topic and still don't understand it. But one thing that I'm wondering if you can comment on based on some of this preliminary work around uterine hemostasis and complement is this concept of local versus systemic complement activity. Because what I find is sort of fascinating about pregnant women is they're in systemic fibrinolytic shutdown. And so, this is, I guess, in particularly commenting on fibrinolysis, but pregnant women are in fibrinolytic shutdown systemically, yet if you sample blood from their uterine blood vessels, it's hyperfibrinolytic, presumably to maintain patency to uterine environment. And so I'm wondering if in any of this preliminary work you can comment on whether there's been any look at sort of systemic versus local complement activity and expression.
Certainly, the data in APLA, the Jane Salmon and others have shown that the abnormalities that occur with antiphospholipid antibodies are definitely local and limited to the uterine. And a lot of it is because the proteins that are important for inactivating or inhibiting complement get dysregulated by, the antiphospholipid antibodies. So there's something that's very local in that disease, and I strongly suspect that if you're going to look for complement's role in, heavy menstrual bleeding or postpartum hemorrhage, it's going to be more at the local level.
Thank you.
A couple of questions, please. One is, what are the known effects of oral contraceptives on complement biology? I mean, you've gone through pregnancy and ovulation related, but when we get into a pharmacologic range, I'm wondering if there's anything known.
Yeah, that's a good question, and I actually don't know all of, I have not looked into that literature other than C4 binding protein goes up with, oral contraceptives and hormonal use and pregnancy, that inhibits protein S activity and makes one more prothrombotic. But that's at a very superficial level. I have not looked actually to see what its role is in.
Because it would be very interesting to see, and of course, we're looking at physiologic effects, but we're also in an era where pharmacologic effects of estrogen-related compounds will play a very major role in women's health with regard to bleeding, potentially. So complement in that setting would be interesting. The other relates to your discussion of inflammation. Now, you didn't talk about any effect on vasodilation or vasoconstriction, presumably vasodilation, but it seems that since that's certainly vasodilation is related to the inflammatory process, and given that complement is playing a role in inflammation, could the uterine environment be really a stage for aberrant vasodilatory activity leading to menorrhagia?
Certainly possible. So the intersection, another major point of intersection between the complement and coagulation system is through kallikrein and bradykinin generation. And so, you have, complement can, through the classical pathway, and can generate, products that, like C1 esterase inhibitor regulates that kallikrein. And so through that intersection, you can have aberrant bradykinin generation that then leads to vasodilation and effects on uterine vessels there. And you also have effects of plasmin on the kallikrein system too, so that could be another point of intersection.
Does bradykinin-related avoidance contribute to menstrual pain?
To pain? Ooh, I don't know that. Yeah, that's a good question.
Maybe.
Very good questions.
Yes. Well, thank you very much, Gal. We asked you to speculate, and we got the audience to speculate along with you. You will,
Very good thoughts. Thank you.
I hope you're now willing to accept the role as the expert of complement and uterine
Yes, there we go.
and you'll be called on again. Thank you
Yes.
very much. Dr. Lawrence.
This is a small community, and one of the objectives of our efforts in this field is to promote integrated efforts with respect to research. And so it would be helpful in that regard if each of the questioners would identify themselves and their institution when they ask their question.
Thank you, Ken.